So we just finished talking about how eukaryotic cells process RNA by removing intronic regions in genes and splicing together exons. The mRNAs are further modified by the addition of a 5' cap and a 3' poly A tail. And these caps and tails will direct the mature mRNA transcript to ribosomes, and they'll also protect the mRNA from degradation in the cytoplasm of eukaryotic cells. Okay, so now that we've talked about this RNA processing, let's talk in a little bit more detail about how eukaryotic cells control gene expression. So we've seen examples of gene expression or gene regulation in bacteria, and we talked about the lac operon, which is a way that E. coli can control whether or not they metabolize lactose. And so it's not useful for them to metabolize lactose when glucose is present, but when glucose is absent and only lactose is present, then they can turn on genes that allow them to import and metabolize the lactose and use it as an energy source. Eukaryotic cells have to meet an additional demand that bacteria do not have to account for. And that is that eukaryotic cells have to create and maintain different types of cells, but they have to do it using the same set of genes. Okay, so what I mean by that is that we can have a muscle cell or a lung cell, for example, and those two cells, a muscle cell and a lung cell, will have the same set of genes, okay? But they may not express or turn on the same set of genes, okay? So they have to have a way to decide which sets of genes they will express or turn into mRNA, and then turn that mRNA into protein. So how do they do this? And the answer is differential gene expression. So what is differential gene expression? So differential gene expression occurs when cells respond to signals from other cells, and as a result of those signals, they express different sets of genes. And these different sets of genes are responsible for forming specialized cells, arranging those cells into tissues, and coordinating their activity in a multicellular society that we call an individual. And so this is a really important process, and we're going to take a deeper look at all the ways cells can engage in differential gene expression. Okay, so gene expression in eukaryotic cells is controlled at many different levels, okay? We have a lot of control happening in the nucleus, and we have then levels of control happening in the cytoplasm, okay? So I'm going to walk through these 6 different ways in which gene expression can be controlled in a eukaryotic cell. And some of them we've talked about before, and some of the ways are new, okay? So one of the new ways in which eukaryotic cells can regulate gene expression is by controlling how tightly DNA is wound, okay? So recall that DNA is wound around these proteins that are called histones, and they're packed in really tightly wound very tightly around histones. And when DNA is wound really tightly around histones, it's not accessible to RNA polymerase. And so this tight DNA protein complex is what we might call a closed chromatin state. And so the chromatin has to be remodeled or loosened so that RNA polymerase can access different promoter regions. And so this open chromatin can then be transcribed. So that's one example of gene regulation, that chromatin has to be opened before RNA polymerase can bind to promoters. So we've also talked about transcriptional control. So recall that in bacteria, sigma binds to the promoter region to regulate gene expression. But in eukaryotic cells, there's a much wider variety of transcription factors besides sigma, and also a wider variety of promoter regions, both in their sequence as well as in their location relative to the gene. Okay, so the last example that we'll talk about in the nucleus is the RNA processing. So recall, right, we just talked about this, The mature mRNA is spliced together, so we remove the introns into the trash, and we keep the exons. We add a 5' cap and a poly A tail. But it turns out that we don't actually have to include all of the exons in a mature mRNA transcript. And so different combinations of exons might be included in the mature RNA, and this process is called alternative splicing. So this mRNA can then move into the cytoplasm, where it can be transcribed, and it can be recognized by a ribosome. But it can also be degraded, and some mRNAs are degraded really quickly, and others have longer lifespans. So the mRNA lifespan varies. So if an mRNA has a longer lifespan, or it's more stable, they're more often translated into protein than these mRNAs with these short lifespans. So whether or not the mRNA will be picked up by a ribosome can also be controlled. And that is controlled by RNA binding regulatory proteins that may bind to the mRNA and enhance the translation or prevent translation. So that's an example of translational control. And then finally, and this is something we've talked about quite a bit, is post-translational control of proteins. So some proteins need to be modified further before they can become activated. So an example of this might be that a protein needs to be glycosylated in the Golgi before it can be fully activated. Another really common example of post-translational modification is the phosphorylation of proteins, which can activate the protein, or it can even inhibit a protein. We've talked about this example when we talked about CDKs that are both activated by phosphorylation and inhibited by phosphorylation. Other proteins can be degraded more quickly by adding a motif called ubiquitin, and this is how the cyclins, which regulate CDKs, were degraded after the end of mitosis, for example. And so there's a lot of ways to wait to turn on proteins or turn them off very quickly. Okay, so we're going to talk a little bit more about these first three steps. Okay, so chromatin remodeling, transcriptional control, and RNA processing in eukaryotic cells, and how these contribute to differential gene expression. Okay, so let's dive into chromatin remodeling. Chromatin remodeling is necessary for eukaryotic transcription. To remind you, chromatin is DNA wrapped around histones, and histones are proteins that are positively charged. And since DNA is negatively charged, they have an electrostatic interaction between each other. And if you look at DNA under a transmission electron microscope, so a very high-powered microscope, the DNA appears as beads on a string. So here is the string, and here are these little dots, these brighter dots being the beads. And it turns out that DNA is very tightly wrapped around these beads. And this level of packing is necessary to fit all of the nuclear material inside the nucleus. If we didn't have the DNA wrapped in this way, we simply couldn't fit that amount of genetic material inside the nucleus. So what are these beads? Well, these beads are called nucleosomes. Okay, so here is a nucleosome. It's a group of 8 histone proteins. And DNA, a DNA double helix, is wrapped twice around this group of histone proteins. And so this is about 200 base pairs, and so this allows DNA to be very tightly packed. But this also presents a limitation, which is that RNA polymerase cannot easily access these genes that are wrapped around these histones. And so there has to be some sort of modification that takes place to allow RNA polymerase access to genes it's trying to transcribe. So first we'll look at some data that suggested that actively transcribed genes were less tightly around histones. And then we'll talk about the ways in which histones can be modified to have this looser or decondensed structure. So scientists were studying two genes in red blood cells. One of the genes was called beta globulin, and this was part of hemoglobin. And in red blood cells, beta globulin is expressed at very high levels. In contrast, the gene ovalbumin is not expressed at all inside red blood cells. And so they took advantage of this nuclease that was called DNase 1. And DNase 1 can act or can degrade the phosphodiester bonds between nucleotides and degrade them so that they're released into individual stretches of nucleotides shown down here. But it can only do this if the chromatin is decondensed. So if it's in this looser structure where it's not as tightly wrapped around the histones and it's not forming what we call fibers here. And so, and again, the DNase 1 can't act on DNA that's wrapped in a fiber, but it can if it's decondensed, shown here. And so what they saw when they did this was that the gene for ovalbumin was less susceptible to DNase, and the gene for beta globulin was more susceptible to DNase. And so what they were able to demonstrate was that chromatin is decondensed in genes that are being actively transcribed, okay? So we have to decondense the chromatin. How do we do this? So there are a few ways in which we can alter chromatin. One is that the DNA itself can be methylated, okay? So here I'm showing a sequence of DNA, and there are these enzymes called methyltransferases that recognize a cytosine followed by a guanine, and it will add a methyl group to that cytosine. And so this is often referred to as a CpG motif in this conformation here where we have the cytosine being methylated. And it turns out that actively transcribed genes have fewer methylated CpG motifs near their promoters. Okay, so that's one way, is by directly altering the DNA itself. Another common way to alter chromatin is by modifying the histones. And this is the one I really want you to focus on. So here we have chromatin wrapped very tightly in a fiber. And you can see they're all clustered together. And an enzyme called histone acetylase, or just shortened to HAT, can actually add an acetyl group, which is CH3COO-, to the histone. So the acetyl group is, because it has this methyl group here, which is neutral, when it's added to the histone, right, it neutralizes those positive charges. And then this here COO-, this carboxyl group, right, it's an acid. And so this will also neutralize that positive charge. And so these become less positive, and that disrupts the interaction with the negatively charged DNA. And so another way to think about this is that these HAT enzymes, the histone acetylases, they can turn on gene expression by neutralizing the positive charges on histones by modifying them. In contrast, there are enzymes called histone deacetylases, and they can remove the acetyl groups from histones. And when that happens, the DNA can wind back together again. And so this will turn off gene transcription. And so here are just two images up here. This is what the fiber looks like. It's quite a bit thicker, depicting that tighter winding, versus when the HAT enzymes are acting on the DNA. You can see it's more of this beads on a string conformation that you saw in the last image. And the final way that histones can be modified or chromatin can be altered is by using these histone remodeling complexes. And these harness ATP to reshape the chromatin. So an example of what a histone remodeling complex could do is it can just simply knock the DNA off of the histones. And so there's free DNA, and RNA polymerase can more easily access promoter regions. So that's what I wanted to cover in regards to chromatin remodeling. The next difference that we'll talk about, or the next way in which eukaryotic genes can be regulated, is transcriptional control. And transcriptional control exists in bacterial transcription as well, but it's more complicated in eukaryotic cells. So in eukaryotic cells, right, we have our transcription start site, just like in bacterial transcription, and it's preceded by a promoter, okay? And so we call this promoter the core promoter. And an example of the core promoter is the TATA box. And I think this is about 10% of all genes have a TATA box sequence for their promoter. An analogous bacterial element that would act as this core promoter would be the -10 and -35 sequences. And when gene transcription starts, right, we have RNA polymerase that needs to be recruited, but first it's recruited by either a TATA binding protein, so if that's the TATA box, this is a specific protein, or what we call a general transcription factor. So again, the take home here is that we have a core promoter upstream of the start site, and it binds to a general transcription factor. And this is going to enhance the transcription of the eukaryotic genes. Okay, but this is not sufficient to trigger transcription. There's also a promoter proximal element. So this is directly upstream of the promoter. Okay, and so an analogous bacterial element for a promoter proximal element is the cap binding site, that's a positive regulator, or the operator that bound the repressor. These are, as I mentioned, regulatory sequences that are located upstream of the core promoter, and they recruit proteins that we either call an activator, or sometimes we refer to it as a transcription factor. And so that's where a transcription factor can bind. So for a long time, scientists thought that we had these promoters and these promoter proximal elements, but it turns out that we have quite a few more sequences that can control the transcription of eukaryotic genes. And this is where enhancers come in, okay? So there's no analogous bacterial element for an enhancer, but these are regulatory sequences located farther away from the core promoter. They can be very far upstream from the promoter. They can be downstream from the promoter, or they can even be within an intron in the gene itself. And so these enhancers can be virtually anywhere. They can actually also flip orientation, so they can flip from 5' to 3', and then flip over to 3' to 5', and this will not change their function at all. And so they also act to enhance gene transcription by binding activators or transcription factors. So they bind to the same proteins to recruit RNA polymerase as the promoter proximal elements, but they're located in different sites. So those are the differences between those two. Not shown in this picture are also silencers. Okay, so these are sequences, that regulatory sequences that are located farther away from the core promoter, and they oppose enhancers. So they decrease transcription, and they do this by binding repressors. So we heard about repressors, right, when we were talking about the lac operon quite a bit, but these sequences are located farther away from the core promoter than in bacteria. Okay, so what's going on here is that the expression, or the sequences of enhancers, silencers, and proximal or promoter proximal elements are helping cells specialize. So most genes are regulated by a combination of these sequences. And there are hundreds of transcription factors that bind to these sequences. So different types of cells express different sets of genes primarily because they have different transcription factors along with having different chromatin structure. So if you take when an organism or an individual is establishing tissues, the genes encoding transcription factors are in turn expressed largely in response to signals that arrive during embryonic development. So you have a signal that says, be a muscle cell, and that then signal then causes the transcription of transcription factors that are specific to turn on genes specifically for muscle cells. So that's how we're arriving at cell specialization in a multicellular organism. The last group that we'll talk about is RNA processing. So we just finished talking about chromatin remodeling and transcriptional control that's happening in eukaryotic cells. And the last step we'll talk about is RNA processing. So just to remind you here, we have a mature mRNA where the introns have been spliced out, and we only have our exons, we have our 5' cap and our 3' tail. RNA processing is rare in bacteria, but it occurs extensively in eukaryotic cells. So there's an additional step of processing that can occur because different combinations of exons can be included in the mature transcript in a process that's called alternative splicing. So let's take a look at what that looks like. So here's a gene called tropomyosin. This is a protein that's important for the function of muscle cells. And you can see that this gene has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 exons. So 13 exons and introns between each of those. So this is the gene that's present in all cells, but alternative splicing will produce more than one mature mRNA from this gene. So for example, in skeletal muscle A, so this is a cell type that's called skeletal muscle A, we'll get a mature mRNA transcript that has spliced together exons 1 to skipping 2, 3, 4, 5, 6, skip 7, 8, 9, 10, 11, and 12, but no 13. In skeletal muscle B, we have all of the same transcripts up until 12 with the addition of exon 13. And so these mature mRNAs actually end up encoding for different versions of this protein that have slightly different functions. And so one gene can encode for multiple polypeptides. And then the tropomyosin gene actually has a lot of alternative splicing transcripts. So in smooth muscle cells, we see the inclusion of exon number 2. In a muscle precursor cell, you can see we have the inclusion of intron 7. And then this protein is further modified, or this mRNA transcript is further modified in the liver and in the brain. So it turns out that over 90% of human sequences for primary mRNA transcripts do undergo alternative splicing. And so the mature mRNAs and proteins actually outnumber the number of genes. And so this kind of throws out the one gene, one polypeptide hypothesis. And now genes are defined as a nucleotide sequence that allows for the production of one or more polypeptides. So I've been going over differences between bacterial and eukaryotic gene expressions throughout this video lecture, but there's a really helpful chart in your book that I just wanted to spend a minute or two going over. So in bacteria, there's very little chromatin remodeling that needs to take place. That's because there's less packaging of DNA, and so remodeling is not a major issue for regulating gene expression because RNA polymerase can access the DNA. But in eukaryotes, there's extensive packaging of DNA, and so chromatin must be decondensed for transcription to begin. Transcriptional control exists in bacteria, and so sigma will bind and interact with the promoter sequences and help recruit, or actually is part of RNA polymerase. And there's also positive and negative control by regulatory proteins that act close to the promoter. And so we have the cap binding sites that bind cap for positive control, and we have the operator that binds the repressor for negative control. But these are all sequences that are close to the promoter. In eukaryotes, we have positive and negative control by regulatory proteins that act close to the promoter as well as far away, right? So those would be the enhancers and the silencers that are far away, and those proximal elements that are close. Binding to these, the core promoter and the enhancers and the proximal promoter elements are a very large set of transcription factors. So instead of having one protein like sigma, we have hundreds of transcription factors that interact with the regulatory sequences or the core promoter. I didn't mention this, but you can read more about it if you're interested. So another protein called a mediator is required to bring together these sequences that are far away and close to the promoter, and so that they can make a three-dimensional structure that's actually now physically close, even though the sequences aren't adjacent to one another. RNA processing is rare in bacteria, but extensive RNA processing occurs in eukaryotes, and we have the alternative splicing of exons by removing both exons and intronic sequences. mRNA stability is rarely used for control in bacteria, and again, something we didn't have time to talk about, but it's commonly used in eukaryotes, and it limits the lifespan of mRNAs. Translation is controlled in bacteria, so regulatory proteins bind to mRNA and ribosomes, and this affects the rate of translation, and that also occurs to some extent, and because of regulatory RNAs as well in eukaryotes. We also have post-translational modifications occurring in both domains of life. In eukaryotes, we also have the addition of short proteins like ubiquitins that can target proteins for destruction. And then finally, for gene organization, in bacteria, genes are often organized in an operon, and so that they can be regulated in a common way. But in eukaryotes, each gene has its own set of regulators and transcribed and processed into mRNA with only one protein coding region. So to summarize what I've told you in this video, gene regulation in multicellular organisms is important for differentiation, communication, and coordination. Eukaryotes can regulate gene expression at many levels. Those that are shared with prokaryotes are transcriptional regulation, translational, and post-translational. And then unique to just eukaryotes are this extensive chromatin remodeling to open up the chromatin, RNA processing to choose which exons need to be in a mature mRNA transcript. And then I didn't talk about it in much detail, but eukaryotes really regulate mRNA stability with these small RNAs, and this is a really cool research topic. So for this video, we focused on more complex transcriptional regulation, right? Turning on specific transcription factors to get specific cell types, on chromatin remodeling and RNA processing. So that's it for today, and I'll see you in class.